Hello, everyone. Our long day is almost over. Some of us have been here since dawn. Glad you're still here. My name's Dave Burdick. I'm the director of the Center on Aging, the Stockton Center on Successful Aging. Appreciate you being here. We've got a few people. First off, thanking all the staff and the great faculty that I work with here, some walking in the room. It's a lot of work, and our staff has done a great job. Brief introduction from me, and then maybe I'll see you after. We're having a little reception in the Board of Trustees room immediately after this presentation. It's my responsibility now, and he's got a meeting in just a few minutes, to introduce Stockton's brand new, he's only been here for 1 week, provost, pretty much the second most important person on campus, maybe the first most for us academics. He's just started here, and he's made a really good impact already. I'm introducing to you Dr. Michael Palladino, who will give you a formal brief welcome from the university before we introduce our keynote speaker for today. Michael, thank you for joining us.
Thank you, Dr. Burdick. Appreciate it. Well, welcome. Good afternoon, everyone. Hopefully everyone is doing well, and despite the long day, and you've had a great, energized day. I really am very pleased to welcome you to Stockton and to welcome our distinguished speaker, Dr. Bohn, leaders of the Notre Dame Club of South Jersey, right, if I have that right, and our faculty, staff, and students who may be here realizing it's an incredibly exhausting time for them at the end of the year now, and other honored guests. Look like you've really had a great day, and hopefully this is a culmination of that day. So as Dr. Burdick mentioned, I've been a Stockton employee now for 192 hours. I started last Monday as provost, but I can't tell you how excited I am to be leading academic affairs at really this truly a wonderful institution. If you don't know too much about Stockton, founded 50 plus years ago with a core mission of interdisciplinary inquiry and collaboration, and it lives it in so many ways, from the design of the facility to our curriculum to the faculty who really want to be here. And in lots of ways, as I think about this, for me, the premise of interdisciplinary work and who Stockton really is aligns beautifully with the topic for today, and also with the new, as I understand it, bioengineering and life sciences initiative at Notre Dame, for which Dr. Bohn serves as the inaugural director, and of course the topic of his lecture today on personalized healthcare and precision medicine. So I would share that I'm a molecular biologist and geneticist by background, and I'm fortunate to be an author for a series of books in biotechnology and genetics that are used by a large majority of colleges and universities around the country. If you go to most schools, 9 times out of 10, you say, do you have this topic or that topic? They're going to use one of those books. And in each of those books, we speak and describe extensively quite a bit about precision medicine, all aspects of precision medicine. So I can't tell you how disappointed I am that I have to go to, I'll say this among friends here, what is going to be a terribly boring meeting by comparison to what all of you are going to have a chance to talk about here, but it was previously scheduled and had no choice. But I'll look forward to hopefully being able to listen to the recording. I do also want to take a moment to thank Dr. Burdick for his work in leading the center, but also congratulate him, as hopefully many of you know, for his retirement next month after 40 years. 40 years on our faculty, 15 years, as I understand it, as our inaugural director of the Institute. And in his last lecture just a couple of weeks ago, Professor Burdick really urged Stockton to continue to embrace and renew its interdisciplinary roots and its background. And I very much appreciated that context, because for me, coming in as a new faculty member and academic leader, understanding that fabric of what's important to Stockton and what makes Stockton unique is truly special. And I heard that a lot during the interview process. So today's lecture, the festival, SCOSA stewardship of Stockton's involvement in a whole variety of things, New Jersey Geriatrics Workforce Enhancement Project that I learned about, that Professor Burdick and SCOSA do indeed talk the talk and walk the walk of interprofessional education, and for that we're very proud. But I also think this shows very clearly the university's commitment to enhancing the welfare of our communities. And hopefully may we all be blessed to be of advanced experience and of advanced biological aging in the future. So finally, I do want to share my congratulations to staff colleagues that I've not even had the chance to spend much time with, to Professors Christine Ferry and Jessica Fleck, who were recently appointed as director and assistant director of SCOSA. I look forward to working with them and other staff as we continue to seek ways to advance the center's success. So I want to, again, thank you all for being here. Thank you for the opportunity to just say hello for a few minutes. I'm sorry I can't stay, but please enjoy the lecture and the rest of the afternoon. Thank you, everyone.
I can tell you, Dr. Palladino is off to a great start. As a professor retiring very soon, I was given the honor as an emeritus professor to process with the platform party at the Boardwalk Hall for our graduation last Friday. And somehow people knew what they were doing, because I've got really terrible peripheral and night vision, but you know, you manage. And so they put Dr. Palladino right in front of me to make sure I didn't steer off course. And they put a minister from the Board of Trustees right behind me. And all the way into Boardwalk Hall, every time we made a turn, he wanted to make sure I didn't turn the wrong way. He would turn around, make eye contact, and like, I'm okay. So in a room like this, I have problems, but in rooms where you're filled with people with black gowns, it's just like dark all over the place, and Michael was very helpful. And the minister was behind me in case I needed any kind of divine intervention. Before I introduce Steve Kluge from the Alumni Association from Notre Dame, I'd like to call out Christine Ferry. Did you leave, or are you still here? Christine Ferry is going to take over the Center on Aging when I retire. She's amazing. I've known her since she was an undergraduate at Rutgers University, and from the State Society on Aging. In the second year of graduate scholarships we handed out, she was one of the recipients. And the foundation was from North Jersey, the Susan Grotta Foundation, and they wondered, why are we paying for a student to go to grad school out of state? She'll never come back. But she went to Duke University, which is no slouch in terms of our field, in fact, some of the world leaders, and she came back. And while I cleaned my office out the other day, I found a really nice note from her mother, handwritten note with her picture, saying, Christine is so happy to receive this award. It means a lot to her. I know that she'll go off to Duke and do a great job, and then she'll come back to New Jersey to be among friends, and that's exactly what she's done. So give her a big round of applause. You'll get to see her quite a bit, and I don't know, do you want to say something or not? She's not gonna talk right now. Let's get the show on the road. So thank you so much. We're so blessed to have such wonderful colleagues here. I'm going to turn things over to Steve Kluge from the Notre Dame Club of South Jersey, who's the sponsor of our Hesburgh lectures. We've had quite a few over the years. This is the first time we've included one in our festival. Steve graduated from Notre Dame. You've ever heard of Rudy? Rudy. They haven't. Well, Rudy, who the movie was made out of, was in Steve's class in 1976. So he's got me by maybe a year. He's an he's an accountant, worked very much for large corporations, including the Educational Testing Service up in Princeton. He is now the club's treasurer. You might be able to buy football tickets from him if you catch him afterwards. He's also regional director of the Notre Dame Alumni Association, and he will have the honor of introducing today's guest speaker. Thank you. Yep, you did. Thank you. I just noticed.
I have to get the light working here. Good afternoon. The Notre Dame Club of South Jersey is proud of our 19-year association with Stockton Center for Successful Aging. This relationship has been nurtured by Dr. Dave Burdick, class of 1983, and the late Elizabeth Elmore, class of 1972. Through their efforts, the Notre Dame Club has been able to host various leaders in the fields of science, engineering, the arts, and business at Stockton's annual Successful Aging Festival. The Notre Dame Club of South Jersey is one of 300 clubs worldwide devoted to supporting the Notre Dame alumni and local communities through community service, continuing education, camaraderie, mentoring, student support, and faith-based programs. And as Dave has said, I also have some football tickets to sell if you're interested. Today, it is the club's pleasure to sponsor Dr. Paul Bohn. Paul, the Arthur J. Schmidt Professor of Chemical and Biomolecular Engineering and Professor of Chemistry and Biochemistry at the University of Notre Dame, was recently named the inaugural director of the new Bioengineering and Life Science Initiative, commonly called BELLS. This joint initiative of Notre Dame, College of Engineering, and College of Science will advance human health and wellness through interdisciplinary biomedical research and training, from fundamental advances to detection, prevention, and treatment of diseases. Paul is an internationally known leader in the field of analytical chemistry. He has served as director of the Berthium Institute for Precision Health since 2008, and also directs the National Science Foundation supported Center for Bioanalytical Metrology. His research focused on molecular nanotechnology, the personal health monitoring, and imaging in microbial communities. He is a fellow of the American Chemical Society, has authored or co-authored more than 300 publications, and holds 9 patents. Paul graduated from Notre Dame in 1977 with a bachelor's degree in chemistry. He then attended the University of Wisconsin-Madison for a PhD in chemistry in 1981. Prior to joining Notre Dame's faculty in 2006, Paul served as head of chemistry department and interim vice chancellor for research at the University of Illinois. Paul has been married to Raylene for 46 years and is the father of 2 children, Nathan and Lauren. Today, it is my pleasure to introduce Paul Bohn, who is here to discuss the future of personalized healthcare through precision medicine.
Well, thank you, Steve, for that gracious introduction. Both thanks and congratulations to Dave. It's a really special honor to be here on this big day celebrating your accomplishments and your 40 years of service to this institution. So I'm delighted to be here. This guy, right in the middle here, Ted Hesburgh, was president of the University of Notre Dame for over 35 years, and in that time essentially transformed the university from a small Midwestern Catholic undergraduate college into a world-class university. He was responsible for probably the single most important event in our institution's history, and that is taking Notre Dame from being an all-male institution to being coeducational in 1972, which coincidentally was the year that my introducer, Steve Kluge, was a freshman student. He probably thought he had hit the lottery. Not only do I get to go to Notre Dame, but there are women here as well. So with that, let me begin talking about what I want to tell you about today, which is this idea of personalized medicine. So to begin, if we talk about personalized, or I'll use the words personalized and precision interchangeably, precision medicine, what is it? Well, it's actually pretty easy to describe. Precision medicine is simply delivering the right treatment to the right person at the right time. What's so hard about that? Well, it's really easy to do imprecision medicine. This table is about 10 years old, so it's a little bit out of date. But basically, for each one of these medications, the red stick figures correspond to the people who are not treatable by that medication, and the blue to the people who are treatable. So things like Humira, not so bad. You get 1 out of every 4 people, but by the time you get to Nexium, it's something like 1 out of 25. Now you may be thinking, why in the world would we waste our time with something like that, until you realize that there really still are a large number of people who are helped by these medications, even though they might be a small proportion of the overall potential patient population. Now, for the last 10-ish years or so, I've been blessed to be associated with what we now call the Bethune Institute for Precision Health. And this group of people that are behind me, these are my colleagues who make up the research and scientific and engineering staff of the Bethune Institute. And basically what the Bethune Institute does is it builds tools. And I'm going to try to convince you today that building tools is a critical component of being able to deliver precision healthcare to the people who need it currently. And in particular, those tools are tools which are geared to understand and characterize human variability at the molecular and cellular levels. And so part of what I want to do today is to try and have you leave with the notion of how different we all are at the molecular level and how that plays out in lots of different features of ourselves. So the Bethune Institute, of course, we focus on developing advanced biomedical technologies. We develop cross-cutting ways of exploring and integrating multiple disease areas. So we're not just a cancer institute, we're not an institute related to metabolic disorders or any other kind of specific disorder. We try to develop tools that can go broadly across all disease and disease classes. And we want to specifically have a real-world impact by creating a community and training young researchers. And even though it's not on this slide, also very important to us is thinking about how to move advances out of the academic laboratory and into the hospital, into the broader society where they can be utilized effectively. Okay, so how precise do we have to be? And I show this slide because I think it really drives home a critical point. In 1999-ish, 2000, we basically solved the human genome. We mapped all of the DNA bases that are responsible for the characteristics that we all display. I put this picture up to show you that we all have about 20,000 genes. C. elegans, which is a worm about the size of my thumbnail, and you can see here in this graphic, it's not a very complicated organism. It also has 20,000 genes. So we all thought that solving the human genome was a big deal, and it was a big deal. It is a big deal still. But why is it that C. elegans has the same quantity of genetic information as Homo sapiens? And what does it mean for us to think of ourselves as these truly evolved and complex organisms, whereas we don't have any more genes than something like a simple worm like C. elegans? Well, I like to use an analogy, which is, DNA is a blueprint. And if you think about blueprints, they're really great because they tell you exactly what to build. And if I think about track housing is based on essentially one generic blueprint. And here you see some homes in a track housing development in Southern California. And at that level, they all look exactly the same. They all have the same housing genome. But if you go inside those houses, they start to look incredibly unique and different. If we look at the one on the left and the one on the right, they're clearly distinguished from each other by the floor coverings, the paint, the furniture that's been put into the house, etc. And so there's way more to the story than just having the blueprint that the carpenter uses to build the house. In the healthcare that we all know and love, we have the same idea, and that idea comes from something we call the central dogma of biology. Now, I'm cognizant of the statement in Stephen Hawking's book, A Brief History of Time, whereby his publisher told him for every equation he put in the book, he lost half of his sales. So I'm going to keep the number of technical slides in this part of the talk to a bare minimum, but I do want to just remind everybody that the central dogma of biology is that our cells contain a genome which effectively consists of chromosomes, and chromosome is nothing more than a physical body that's colored. That's why they're called chromosomes. And those chromosomes are composed of highly compactly organized deoxyribonucleic acid, or DNA. And for years and years and years, we thought that what happened was that every DNA molecule uniquely coded for the fabrication or synthesis of a particular protein. So one gene went to one protein. And then the proteins we know, and we have known for quite some time, they're responsible for, either individually or in multi-protein complexes, they're responsible for performing many of the functions that cells perform in our bodies. It's a little problem. And the problem is that we know that every cell has about 1 million proteins. All right, and if we have, I use 25,000 here rather than 20,000, so the actual exact number doesn't really matter, but you can clearly see that if you start off with 25,000 blueprints and you produce 1 million different parts, there had to be something between the blueprint and the production of the part, in our case the protein, which is responsible for this variability. And that is called generically the proteome complexity. So if we start off with 20 to 25,000 genes, well, first of all, we know that those genes aren't uniformly translated into messenger RNA. So there are alternative promoters, splicing, editing, etc. So for every gene, we end up with something like 4 or 5 different transcripts. And then those transcripts are converted into proteins in the ribosome and they're converted through something called post-translational modification. Now, this is all the technical goo. What you really need to understand is that I start off with one of these things and I end up with about 40 of these things over here that really matter. So just by solving the human genome, we haven't solved the problem of human variability and complexity as it relates to healthcare. Furthermore, just to reduce my sails even further, I'm going to show you one more technical slide in this part of the talk. If this is one protein which is encoded by a specific gene, and this is the same protein ostensibly encoded by the same gene, you can tell by looking at this picture that they're different. So this little guy has a little black squiggle there, and this has a red arrow, and this little guy has nothing here, and this little guy has a little yellow dot. And so these are just chemical modifications that occur as your DNA is being translated and then used to produce the proteins that perform the functions in your body. We can take all of that into account and just ignore this stuff here in the middle by saying that every protein that's encoded by a gene is not really a single protein, but it's a whole family of what are called proteoforms. This particular family has 8 members, and they can have up to, as we saw, they can have up to 40 or 50 different variations, but this one we've just used for purposes of illustration has 8. And then furthermore, these all fall into networks. So the function of a protein frequently is to take something which is produced here, change it, and release it over here. Much like, and I'm sorry to use a Notre Dame football analogy, but much like the quarterback takes the football from the center, the quarterback very rarely, or at least used to very rarely, run with the ball himself. The quarterback either turns and hands it to somebody else to run with it, the halfback, or throws it to another individual, maybe a wide receiver. So the quarterback is very much like one of these proteoform families, which takes a chemical substrate and then changes it and hands it off to the next member of the molecular football team here. Okay, so hopefully we're going to now go to the fun part, which is what are we doing at Notre Dame and why does it matter? Well, the first thing I'm going to tell you is that technology matters. And the reason that technology matters, I think, is illustrated in the following few slides. First of all, this is my colleague, Norm DeVicki. Norm was recognized as one of the unsung heroes of the Human Genome Project by the premier journal in our field, Science. And basically what they recognized him for was developing a sequencing technology. What that means is that DNA consists, as you know, of 4 different kinds of nucleotide bases, and we call them A, G, C, and T. And what Norm and his colleagues, his students, developed was a way to actually read off in real time A, G, A, C, T, G, G, etc. So he could take a string of DNA and basically tell you exactly what its chemical composition was. And he did that by developing some truly amazing technology in which the DNA was pumped through capillary tubes and was read off by shining a laser on them and looking at the various colors which were developed. I will tell you that that technology is now completely obsolete. So everything that you see or read about in the papers that's done over the last 5 to 10 years and into the future is done by something called next-gen sequencing. And the big difference is, instead of looking at one base at a time, now we can look at thousands and thousands of bases simultaneously. So one of the things that you would like to know is you would like to know more about this relationship between the genome, the information that's encoded in your DNA, and the proteome, that is the information that actually results from the transcription and translation of your genetic information into peptides and proteins. And that is done by a series of tools, and I'm not going to go into telling you what these tools are, but essentially they take a complex goo or soup of these different protein molecules, and they, first of all, Norm and his colleagues separate them. So when you see the word chromatography, you can just think of that like a piece of filter paper, and you put something on it and molecules will move in that filter paper at differential velocities. So every one of these peaks that you see in this diagram corresponds to one or more chemical compounds or one or more proteins. And then that's where the field was for a good 15, 20 years. And then Norm and his students and others decided that, well, wouldn't it be really great if we could take one of those peaks and actually pull it apart and see what was inside of it. So they do an experiment called mass spectrometry, or MS, and they see these big peaks that are all associated with this single peak in the chromatogram here, and that tells you that this is not just a pure chemical compound, but it's actually a mixture of different compounds. And then they furthermore take this and break each one of these, the red, the green, etc., into their component parts by doing a second round of mass spectrometry. I'm not going to walk you through this, but the basic idea then is that by putting all of this information together and by using advanced tools of bioinformatics, it's possible to actually identify the proteins. So when I was talking a minute ago about the proteins being different than what you would have predicted based on the genome, this is how we know that they're different. We know they're different because we've measured their molecular mass. Okay, well, that's fun, and that's clearly technological. I think we would all agree about that. Now, the next thing I'd like to ask you is this question: what is this microbiome I've been hearing all about? Well, the microbiome really results from the realization that all of us are not really human beings. Well, we are human beings, but we're much more than human beings. And what we are is we're actually traveling microbial zoos. So associated with our digestive systems, our mouth, our skin, urogenital systems, we have and we carry with us a unique cadre of microbes, bacteria mostly, but not entirely, unique cadre of microbes which determine the overall biomedical status of our organisms. Now, we know a lot about the human microbiome. We know probably 0.00001% of everything there is to know. So what we know is we know how many phyla we've looked at so far, the number of genera, species, and we've identified about 300 million genes. Remember I said that the humans have about 20,000 genes, so this is about 15,000 times larger than the number of human genes, and that's because we have so many different kinds of microbes that decide to lodge themselves in our bodies. And this diagram is really just showing you that in some cases we actually can go and we can map those, but in many of these other cases, the ones here in gray, we know really nothing at all about the genetic diversity of the organisms that are resident within those particular arms of the diagram. All right, so how can we get information about what those microbes are and what they're doing? Well, pretty clearly, you know, if they're microbes, we're going to need the tools of microbiology. And if they're molecules, we're going to need to know something about molecular biology and biochemistry. And so we need some really smart people to do all of that. And Matt Champion is exactly one of these smart people. He's my colleague in the Department of Chemistry and Biochemistry at Notre Dame. And what he decided to do is, well, if separating molecules using chromatography was such a great idea, why don't we try to do the same thing with microbes? Now, that may not sound so revolutionary until you understand that a microbe is some 9 trillion times larger and more complicated than a single molecule. So being able to do this was no mean feat. So they developed some tools, and here you can see, starting off with something like a plate where you would grow up individual colonies of bacteria, and then they were able to sample those bacteria using a molecular speciation tool, and then feed those into one of these next-gen sequencing instruments. And you don't have to know what these colors mean. I just want to point out that the genetic complexity associated with each one of these different wells is represented by the color scheme here, and what you can see is that they are all very different. All right, so what that tells us is that as we go from one kind of bacterium or one kind of microbe to the next, to the next, to the next, we're not just changing the identity of the microbe, we're changing its entire genetic makeup. One of the other things about bacteria is that they like to hang out together. And if you know anything about, you may have friends who are microbiologists. I have a friend who's a microbiologist, Josh Schrout. Wonderful friend and colleague at the University of Notre Dame, and this is from his laboratory. And what you can see is that the bacteria shown in this movie are evolving in time to create colonies which have very specific spatio-temporal characteristics as given by these arms. And I'm just running this movie back and forth, so there's nothing special about the reverse part of the movie. The issue here is that you're looking at the entire collection simultaneously. And if you measure the properties of that collection, it's kind of like, and my apologies again for those of you who are not Notre Dame people, but it's kind of like going in to Notre Dame Stadium on a particular Saturday afternoon in the fall and looking up and seeing 80,000 crazed lunatics in the stands. And all you can really say is, well, yep, there's 80,000 crazed lunatics, I mean Notre Dame fans, in the stands today, but you don't really know anything about them individually. So in order to learn something about them individually, Josh and Josh's students and my students developed this really cool device which allows us to individually capture a single bacterium, but not one bacterium at a time because there are thousands and thousands of them. We can capture whole arrays of single bacteria in these little plates. And when we do that, and I'm not going to walk you through these experiments, but I'm just going to tell you that if you perturb one of these samples with a perturbation that looks like this in time, and you look at the entire array, this is the response that you see. But if you look at these individuals, you see that some of them do nothing at all. They're completely immune to the perturbation you're applying. Some of them, like this yellow one here, it pretty much follows the behavior you see for the entire population. And then some of them basically just are kind of what I call the psychotic outliers of the population, shown here in purple, and they display these incredible bursts of activity at very, very specific times. So the population that you have in microbiology is not the same as the individual microbes that make up the population. And that was the key finding from Josh and his students. One of the other things that you have to be able to do in precision medicine is you have to be able to make medicines. Okay, that sounds trite, but the problem with, well, there are many, many problems with making medicines, as I'm sure many of you in the audience know. But one problem is actually getting the medication to to the right location. If you take a pill or you get a shot, you're basically injecting that material into the body at a specific point in space and time. There's no guarantee that 99.999% of it won't just be wasted. In fact, it is in most cases. So what my colleague Bashar Bilgisser in chemical and biomolecular engineering decided to do was to build a car, essentially a vehicle, for the medication so that the medication only went to the correct place in the body. And without going through all of the details here, I'll just point out this little red circle here is the target peptide, it's the targeting element of the peptide lipid conjugate, and this is recognized by these little cup-like structures on the surface of a cancer cell so that it's only directed, the medication is only directed to the cancer cell and not to any of the other cells where it won't have any effectiveness. And you can see here, if you do nothing at all, the gray line, the tumor volume grows. So in this diagram, up is bad and down is better. If you put just the drug in, that's the blue line here, you get a dramatic improvement. But if you put the drug in one of these targeting vehicles that Bashar and his colleagues have made, you get a much better response. So the double little asterisk there means that this is better at a 99% confidence level. Well, that's great. So we can deliver drugs to tumors on demand, or at least we can do that in some cases. What about inside the tumor? Are they all going to the same place in the tumor? And that becomes important in some cancers. And so it's important to begin to be able to pull apart the way that drugs are delivered to tumors. And my colleague Kai-Yu Fu thought about this for a while, and he thought, we can go inside of a tumor and make measurements inside the living animal. Now, he hasn't done this on humans, so these are all mice studies. But the idea that he came up with was building a simple electrode on a needle. And the electrode has in this particular case, has 3 different channels. And so each one of those channels, because it resides on a different place in the needle, will actually be sampling a specific location within the tumor so that you can learn something about the way in which the drug is effective, but not only that, how its effectiveness changes as a function of space, where it is in the tumor, and time. So if you look at these curves here, you can actually begin to do pharmacokinetics. And if you look at the mean or average behavior, the pharmacokinetics is very different than the pharmacokinetics that you see at these individual locations. So again, being able to home in and be very specific and precise in your measurements is incredibly important. Next, we're going to look at imaging. And the reason that imaging is important is, frankly, putting electrodes into tumors is great. We can get information we could never get before, but wouldn't it be nice if we could just take a picture? Because pictures carry an incredible number of independent, parallel channels of information simultaneously. And Tom O'Sullivan, my colleague in electrical engineering, decided to do this in a very specific application where he was looking at the behavior of oxygen molecules in various biomedically relevant situations. And I'm just showing you this slide to show you that, it's not like he went into the lab one day and told a graduate student to do an experiment, and the experiment worked, and so he published it. No, he had to think about all of the steps associated with this novel tissue imaging process and development, starting from system integration and the development of the actual instrument, which is shown here. Obviously has to be made using the tools of microfabrication. He worried about optoelectronic device design and fabrication. And so here you can see some of the materials and structures that are responsible for the emission of the light in these structures. He had to worry about the interaction of the light with tissue, so there had to be some tissue diagnostics. And then he had to worry about signal processing and data visualization. As you see here, the tool was, in this particular case, was used for the development of, or the assessment of the treatment of breast cancers. And then finally, after you've done all that work in the laboratory, you think, oh, great, I'm done. But no, you're not really done. You're not even close to done. You have to show that it's effective and can be used in organisms. So there's technology validation, and that's typically done with animal studies. And then if those are successful, then you can go on into human clinical trials, which I'm happy to say have been very successful. In his case, a very, very effective method of looking at the effectiveness of various treatments for breast cancer and for the development of the initial cancer itself. Well, this was such a great idea that he's now taken it and done many, many different things with it. So it's because it measures molecular oxygen, you can do tissue oximetry, you can use it as an adjuvant to chemotherapy to see if the chemotherapeutics are working effectively. You can use it in perinatology to do imaging of neonates. And then the original application was breast cancer diagnosis. I'll just say parenthetically that Tom's wife is a breast cancer survivor, so this was something that was not only important to him as a scientific and engineering problem, but it was something that was important to him as a human being and as a spouse. And then finally, brain imaging, and they've launched a company, Nearwave, to take all these applications and to move them forward. Okay, that was fun. Well, can you do anything else with imaging? Yeah, you can do lots of things with imaging. This is just one really cool example. This is my colleague Rob Nierenberg, and Rob is concerned about the problem of bacterial or antibiotic resistance, or the resistance of bacteria to the antibiotics that we use as life-saving drugs. And he had a theory that in some cases the antibiotic resistance would result from the formation of what are called biofilms. And biofilms are thick polymeric structures which are produced by bacteria on surfaces, and they exist, they're very complicated chemically, and they exist to do a lot of things, but one thing they do is they protect the bacteria from agents in the outside world like antibiotics. So he was worried about how could we break up these biofilms, and in this movie, green is bad because those are the living bacterial cells, and red is good because those are the dead ones. And so after we start the ultrasound, what you can clearly see is now the red begins to really drown out the green, and we can go from a very robust, resistive biofilm to one that's really been compromised so that our antibiotics can get in and do their job. Okay, last but not least, I'm going to point out some really difficult problems, because these problems being pursued by my colleague Maria Lieberman are basically intended for work in the developing world. Now, why is that a problem? Well, the developing world, often you don't have reliable power. The lack of reliable power means you don't have what's called a cold chain. You don't have the ability to keep reagents functional and alive. And so Maria has basically developed the capacity to do analytical chemistry on a piece of paper. These are called paper analytical devices. I'll just show you two examples. One shown here is looking at the presence of counterfeit drugs. So if you're in sub-Saharan Africa, many of the things that assail you can be essentially treated if you can just get the right medication. Unfortunately, counterfeit medications have become a big business in Africa and other parts of the developing world, and so it's important to be able to tell the good drugs from the fake drugs. And Maria was able to do this using a very simple paper chromatography panel. So you can see here on the left-hand side, the good dosage form. This green box essentially shows this long black line, whereas the same lane, the F lane over here, shows this nice bright blue. And we know from preliminary studies that tells us that that drug is not the effective form of the dosage. She can also do a very crude kind of genome mapping or genetics by looking at the colors that are developed using reagents in these papers, and then advanced tools of machine learning and artificial intelligence in order to classify these into the various classes associated with the development of the DNA structures. Okay, so I went through a lot of stuff, and you might be thinking, well, okay, that's great. That's all in the laboratory. Can any of this actually be used for something useful? Well, I don't know what things were like here at Stockton in 2020, but I can tell you at Notre Dame they were crazy. Like many other universities, like most universities, we sent all of our students home at spring break, March of 2020. And we sat around for the next several months and tried to decide what to do when the students came back. And we developed a plan. And the plan had some good parts. I don't want to completely pan the plan, but the plan had some fundamental flaws. And one fundamental flaw is that they forgot they were dealing with 20-year-old college students. So the plan was we would have everybody come back on August 10th, and then we would do the entire semester and end the week before Thanksgiving. We would not do our normal fall break that we normally do, and there would be no football games, et cetera, et cetera. Well, we did all of that, but the thing that the university officials who were devising the plan forgot to take into account was the fact that these people hadn't seen their friends in 6 months. And so the first thing they did when they got to campus, they all went down to Eddy Street and they partied like crazy, probably mask-free. I don't know for sure, but I'm guessing. So big problem. We had just a wildfire of COVID outbreaks, and the university president, Father Jenkins, was literally 12 hours away from sending everybody home. He talked to Mark Fox, the director of the St. Joseph County Public Health Organization, and Mark said, why don't you just give it another chance? But in addition to the things that you're doing, why don't you institute a program of surveillance? Okay, so this is my example of how you can apply this. This is what I did every 2 weeks for about a year. I would go over to the Joyce Athletic and Convocation Center, which was the old hockey arena, and this is what it looked like on the inside. You had some stations, so you'd go in, and the first thing you would do is you would show your ID that would basically say who you are, and then you would go to a table where a nice lady would hand you a vial. All right, now the reason the vial is important is because you remember back when COVID first started, you would get tested by going to a place and somebody would take a long swab and stuff it as far back into your nose as they possibly could and try to kill you with it. Just kidding. But it was very, very unpleasant experience, and the idea that we were going to get college kids to come and do that once a week was pretty far-fetched. So we developed this test based on saliva. So the saliva would be collected, and it's just what it sounds like. You would stand there in the middle of the hockey arena, and you would be spitting into a tube until you got the right volume, and then you would cap it and hand it to another person, and they would take it, move it over to the genomics laboratory, and then we would run these RNA-based assays. The reason I'm telling you all of this is that this was all made possible by a unique development of one of our alumni, Paul Hergenrother, who's a chemistry professor at the University of Illinois at Urbana-Champaign. And Paul developed this saliva-based assay, which was important because it got around problems associated with various kinds of enzymes that would break down the mRNA and invalidate the tests. In addition, well, that was great. That was certainly great to be able to have the students stay on campus and not have to do remote learning for another year. But we began to think, having everybody show up once a week, that's a lot of bother, and not everybody was compliant. So wouldn't it be better if we would only ask the people who were at risk to show up so they could be tested? And to do that, you have to be able to look at populations of people simultaneously. Well, one way to do that is to take advantage of the things that we all do every day, and no exceptions, and that, of course, is to poop. Okay. So if you sample the waste system, the wastewater that comes out of a building, in our case a college dormitory, you can immediately tell, just by doing genomics at that level, you can immediately tell if the test organism, in our case the SARS-CoV-2 organism, was present in that test sample. So if it's present, then you've got to go and test everybody individually. But if it's not present, you don't. So you realized an enormous savings of time, energy, and money. And my colleague Kyle Bibby in mechanical engineering was responsible for developing that test. Okay, so finally, just to end here, where is all of this headed at Notre Dame? Well, At the beginning, you heard a little bit about this new bioengineering and life sciences initiative, and the idea is really as simple as it sounds. Wouldn't it be great if we could now take the kinds of things that we've been doing individually and begin to integrate them across the entire campus? So going from people who worry about science to people who do engineering, and then ultimately to look at the impact that we have on human health. BELLS, the Bioengineering and Life Science Initiative, will be an activity that's it's kicking off. We started it in February of this year. We're developing teams that are multidisciplinary, so we have mechanical engineers working side by side with molecular biologists. We're investing in infrastructure, bringing new powerful microscopes to campus, recruiting new faculty who have different skill sets and are importantly invested in this idea of collaborating, new programs for graduate and postdoctoral training, and then importantly, developing partnerships outside of the university that will allow us to amplify this message as we go forward. Well, with that, it's been a wonderful time to be able to spend here with you today. I want to, again, say congratulations to Dave and especially to the Stockton Center for Successful Aging, SCOSA. Wonderful idea, and I think one that you can be justly proud of, and to thank also the South Jersey section of the Notre Dame clubs. And with that, thank you. I'll be happy to answer questions if you have any.